Okay, now let's study about the sliding filament theory. This is the accepted model of the striated muscle contraction. When a skeletal or a cardiac muscle cell get contracted, you all know that the thick filaments, that mean the myosin, and the thin filament, the actin, myosin and actin, in each sarcomere slide will pass each other. Okay. While passing each other, what happens? They pull the Z line at each end of the sarcomere closer to one another. What happens? Then the sarcomere gets shortened. So I said y'all that only the sarcomere will get shortened. That means it will reduce its length, but the filaments will remain constant. Okay, they will remain relatively constant length. So the myosin is the motor protein which does the muscle contraction, which will pull the thin filaments towards itself. Okay. Each myosin composed of a head region and a tail region. The tail region is bounded to the thick filament while the head region sticks out. And the actin molecules, that mean the thin filaments, also have binding sites for head region of the myosin molecule. Yeah, it have binding sites for head region of the myosin molecule. That means the myosin head can be bind on the actin filament. When the myosin is at the low energy state, or else the thick filament is at the low energy state, they can bind with an ATP. Okay. While this ATP hydrolyzes to form ADP and inorganic phosphate by releasing energy, this myosin head will enter to the higher energy state. Now what happens? This myosin head will bind to the actin filament. Yes, this myosin head will bind to the myosin binding site in the actin filament, or else in the thin filament. How they get attached? Using a cross bridge. Yes. And then they will release the produced ATP and inorganic phosphate, while then the myosin head will return to the lower energy state. At that time, the thick filaments, that means the myosin, will pull the thin filaments towards itself. Or else you can say the myosin will pull the thin filaments toward the sarcomere. Then what happens? This sarcomere gets shortened. And when a new molecule of ATP binds to the myosin head, what happens? What about the cross bridge? Yes, it gets broken. Then what happens? This myosin head detached from the actin and a new cross bridge cycle begins again. This is a cycle. It happens repeatedly. The contractions of our muscles needed many number of repeated cycles of binding and releasing. Each cycle of myosin head which released from the cross bridge and newly bond ATPs hydrolyze, which promote the binding site of the myosin again to a new actin molecule. This process occurs along every microfibers in the muscle cell. So when the thin filament has moved towards the center of the sarcomere, a new binding site for the myosin head region is exposed in the thin filament. That means exposed in the actin filament. Many myosin filaments can found in one thick filaments. Okay, that means within one thick filaments, many number of heads and many number of tails. Within a second, you can form these cross bridges and you can break all these. So what about, have you all heard about calcium 2+? Yes, the calcium 2+ and some other proteins also plays a major role in the muscle contraction. So guys, that's all about the sliding filament theory.